Our project discusses cancer immunotherapy by Elizabeth Barnes, Hannah Gray, John Wang, and Alyssa McDonald.
Clinical significance. Cancer affects people worldwide both directly and indirectly. There are 7.4 million deaths worldwide due to cancer. The death rates since 1991 have decreased thanks to current forms of treatment, but they all still have problems. Chemotherapy is systemic, so it reaches the whole body, but it does not target cancer cells, so it often kills healthy cells as well. Radiation is not systemic, so it only targets one specific region, leaving cancer to spread to other areas. Surgery is also not systemic and can be dangerous for certain areas of the body like the brain.
So what is cancer immunotherapy? Cancer immunotherapy is a type of cancer treatment designed to boost the body's natural defenses against cancer. Cancer immunotherapy works in the following ways. It stops and slows the growth of cancer cells. It stops cancer from spreading to other parts of the body. It helps the immune system work better at destroying cancer cells. So there are several types of immunotherapy. First is non-specific immunotherapy. It indirectly targets cancer cell, and there are two common types of non-specific immunotherapy, and they are: one, interferons. These cells are made in laboratory, and they generally help the immune system fight cancer and slow the growth of cancer cells. Some common interferons used are wolfram A, intron A, and alpha. Second common type of non-specific immunotherapy is interleukins. Common interleukin 2 or aldesleukin produce cells that destroy cancer. There are some side effects, and there are low blood pressure, weight gain, and other that can be treated with current medication. Second immunotherapy is oncolytic virus therapy, and it is genetically modified cold store viruses called T-VEC, and they are injected into the body, which enters the cancer cells to kill cancer cells. There are some side effects, and they are fever, nausea, and fatigue. Third immunotherapy is called T-cell therapy. T cells are removed from the human body and modified to have a specific receptor, and these receptors allow T cells to recognize cancer cells. These T cells proliferate and are injected into the patient's body to destroy cancer cells. This therapy is specifically called CAR T-cell therapy. Fourth type is monoclonal antibodies, and this is generally it targets therapy is a targeted therapy to block protein in cancer cell, and more details will be covered by Beth.
Monoclonal antibodies attach to certain antigens found in cancer. They recruit the body's immune system to fight and attack harmful cells. They have been found to inhibit cell growth by decreasing antigen expression in proliferating cells.
Is an emerging field of science at the interface of biology and engineering. Mechanobiology focuses on the way that physical forces and changes in cell or tissue mechanics contribute to development, physiology, and disease. While medicine has typically looked for the genetic basis of disease, advances in mechanobiology suggest that changes in cell mechanics, extracellular matrix structure, or mechanotransduction may contribute to the development of many diseases, including atherosclerosis, fibrosis, asthma, osteoporosis, heart failure, and cancer. Cancer treatment failure is invasion and metastasis, and invading tumor cells utilize many of the motility patterns that have been documented for normal morphogenesis. Recently, the role of mechanical forces in guiding various tissues and cell movements in embryonic development has been systematically analyzed with new experimental and computational methods. The tissue and cellular mechanobiology approach also holds promise for increasing the understanding of tumor invasion. The mechanical stiffness of tumors has correlated with invasiveness and manipulation of extracellular matrix, ECM, stiffness in vitro has suppressed the cancer phenotype. All these observations suggest that a synthesis of the biology of cancer cell invasion and cellular mechanobiology may offer new targets for the treatment of malignant disease.
Myeloid leukemia, or AML, is currently the deadliest form of leukemia in the United States and is responsible for over 10,000 deaths per year. Part of the reason for this mortality rate is that AML is significantly more common in the aging population and is known to be caused by treatment of other previous cancers, exposure to certain pesticides, and smoking. AML is distinguished by mutated myeloid stem cells that do not differentiate correctly into red blood cells, myeloblasts, or platelets, and instead amass in the bloodstream. In 2015, scientists discovered that by treating AML cells with certain antibodies, they could force the cells to mutate into other kinds of cells. They were especially inspired when they discovered that some cells transformed into natural killer cells, a form of white blood cell that is integral to the immune system. Furthermore, these new killer cells appeared to target the unchanged AML cells. Thus, the scientists named these antibodies fratricidins. These new killer cells are characterized by long filopodia protruding from the outside of the cell that force nearby cells to lyse or break. The picture on the slide depicts a transformed AML cell in the process of attacking another cell, indicated by the arrow. One interesting fact about the transformation of these cells is that natural killer cells are not innately derived from from the myeloid cells. Instead, they are derived from a different stem cell in the bloodstream.